Well, it's 5 after, and like to go ahead and welcome everybody to the summer edition of Reaching Out While Locked In. We, at the University of Kentucky Beef Extension Service, we found that our seminars, our webinars during the COVID pandemic have been pretty successful and very popular, so we thought we'd extend it into the summer. But we're taking a little bit different slant this summer than what we did during the spring. Our researchers in the Department of Animal and Food Sciences have very little exposure to our clientele, and our clientele have limited access to the research that's occurring in our department in every phase. And so tonight we really begin our investigations or our explanations maybe of the research that is occurring in the Department of Animal and Food Sciences. Tonight we'll focus on reproductive physiology, and there'll be a short combined lecture between myself, Dr. Les Anderson, and Dr. Phil Bridges, who is a reproductive physiologist in our department and teaches the reproductive physiology class and has a very active research program. The focus of tonight is going to be factors impacting fertility, and Dr. Bridges is going to discuss in detail the research that he's doing with the selenium and the form of selenium and how it impacts conception rate and fertility. My role will be simply to facilitate and then kind of give a little bit of an introduction to make sure that everybody's kind of on the same page with fertility and some of the factors that Dr. Bridges is going to talk about. Phil, you want to go ahead and advance the slide? When we talk about fertility, I mean, I get these questions all the time. You know, doc, help me improve the fertility in my herd. What's wrong with the fertility? And one of the problems that we have with fertility is we don't have normally the same approach or the same definition for fertility between people that are trying to communicate, between a researcher, an extension specialist, and a beef producer. And so I thought it'd probably be best if I just throw out what some of the terms that we use when we try to define fertility, so that everybody's on the same page. And when Dr. Bridges gets into his lecture and his description of the impacts of selenium on fertility, you'll be able to recognize differences between conception rate and pregnancy rate. And so I've listed here 4 different terms that are used to define fertility. The first is conception rate, and conception rate is the number of pregnant cows after a single estrus divided by the number of cows bred. So if you had 20 cows that were in heat and you bred all 20 of them and 10 of them conceived, your conception rate is 50%. But that's at 1 estrus, okay? Pregnancy rate is the number of cows that conceived at the end of the entire breeding season. So it'll be multiple occurrences of estrus, okay? And that's divided by the total number of cows exposed to the bull. I've got here total number of cows, but it's the total number of cows that were exposed to the bull. So let's say you turn the bull in on May 20th and you remove the bull on August the 1st. You've got 20 cows. When you pull the bull, you had 20 of them pregnant. Your pregnancy rate is 100%. Okay? And so conception at each estrus must have been high because your pregnancy rate ended up being 100% at the end of the breeding season. And then, of course, slightly less used terms for fertility are listed below. One's calving rate. That's the number of cows that calved divided by the number of cows that were exposed to the bull at breeding. And then weaning rate, which is the number of calves that are weaned divided by the number of cows that were, again, exposed for breeding. And so those are the 4 major terms that we use just to define fertility. That doesn't really look at efficiency. We've got a lot of other calculations we use to define reproductive efficiency, but these are the main terms that we use to define fertility, and the 2 that we really need industry terms that we really need to pay attention to and kind of differentiate is the difference between conception rate and pregnancy rate. Dr. Bridges, if you'd advance it, please. So what are some of the factors that impact fertility? Okay, what are some of the management protocols or management tools that impact whether a cow can conceive and remain pregnant? I hate to bring this up because I know Dr. Van Valen and Dr. Lindcooler are both listening, but nutrition is king when it comes to reproduction. And I need to throw Dr. Bullock in here just a little bit because nutrient availability and nutrient status of the animal kind of go hand in hand, and genetics certainly play a role in the animal's ability to survive and thrive on the amount of nutrients that are available. But nutrients and nutrient availability are they are the key to fertility. Our goal is to get a cow in a body condition score 5 and to maintain her at around a body condition score 5 to 6 pretty much throughout her life, her reproductive life. I don't mind if they cycle down just a little bit, as long as when they get ready to calve, they're somewhere between a body condition score 5 and 6, and we do the best we can to maintain that body condition score through the end of the breeding period. Remember, body condition score is just a visual estimate of how much fat's on the animal's body, and what that gives us is a very raw indication of how that animal is processing nutrients. So is it getting enough nutrients, and does it have enough nutrients in its body that it's storing fat versus mobilizing fat? And of course, the nutrients that are important, and we're not going to lecture about nutrition tonight really, but energy and protein are the two that we pay the most attention to. We have to have adequate energy. We have to have adequate protein for that cow to maintain a positive nutritional balance. But one thing that we don't pay enough attention to is minerals and the availability of macro and micro minerals in the animal's diet. And that's really kind of the focus of what Dr. Bridges is going to talk about, is some very in-depth discussion on the form of mineral and its impact on reproductive physiology and the ability to get cattle to conceive. Hard to really separate nutrition and health much, because if you don't have an animal that has adequate nutritional status, including mineral status, it's very difficult to have a healthy animal. The cow's ability to mount an immune response depends upon her nutritional status, including mineral status. And so we have to start with nutrient availability and nutrient balance and body condition score, and then make sure that we vaccinate those animals and, against respiratory disease complex and lepto, to make sure that those cows don't abort. But again, it starts with nutrients, nutrient availability, and nutrition, and then everything kind of flows down from there. Now, one thing that we have here in Kentucky that has a tremendous impact on our conception rate and pregnancy rate is our environment. We're in the middle of the fescue belt, and in our fescue we have endophyte, which is a fungus that is symbiotic with fescue, and that fungus produces alkaloids, which stimulates heat stress in our animals. Tons of research that have shown that conception rate, or again the ability of a cow to conceive at one particular estrus, is cut in half when cows consume endophyte infected fescue and are subjected to tremendous heat stress. We've done a lot of that work down in Princeton, but it has been replicated, all over the all over the fescue belt. Heat stress certainly reduces pregnancy rate. It certainly reduces conception rate. And animals that consume endophyte infected fescue during the summer months are heat stressed to the point where their conception rate often does not get to 30%. Okay, and so let's think about that number for a minute. Our normal conception rate is somewhere between 60 and 70%, but during our heat stress moments, our conception rate drops to 30% or lower. And so if you just do the math, that's 7 estrous cycles to get more than 90% of your cows pregnant. And that is a tremendous impact on our ability to get cattle, for cattle to conceive and to get our cattle pregnant. And actually, I think, I know Dr. Limcooler talked about it a little bit a few seminars ago, and Dr. Bridges may touch on it a little bit tonight, but our mineral content certainly impacts the ability of our cattle to cope with some of the endophyte infected issues with heat stress. And so again, the minerals can play a huge role in our ability to get cattle pregnant. And so with that, I'm going to conclude the very short extension portion of tonight. Of course, I'm going to be interjecting some during Dr. Bridges' presentation, but I'd like to introduce our featured speaker tonight, and that's Dr. Phil Bridges. Dr. Bridges is actually a native of Australia. He grew up on a sheep station in western New South Wales in Australia, and he and I were sharing some stories about shepherding in Australia and some of the unique environmental and different challenges of Australian agriculture compared to the United States. Phil got his master's degree at the University of Hawaii with a fairly well-known reproductive physiologist, Charlie Weems. He then went on to West Virginia and studied, got his doctoral degree with Keith Inskeep, who's considered one of the godfathers of reproductive physiology. And Phil doesn't know this, but I met him back when he was at West Virginia. He was given a seminar at the Animal Science Association meeting while he was there, and he and I met each other back in those days. Dr. Bridges then went to Cornell in New York and got to his postdoc with Joanne Fortune, who he's going to share some of Dr. Fortune's research tonight. Her main goal was to elucidate how follicles grow and how that impacts fertility in cattle. And it was truly groundbreaking work that she and her lab did there, and Phil certainly was a part of some of that research. Dr. Bridges came to the University of Kentucky in 2006. He actually was a member, he joined faculty over in the UK Health Sciences department and did clinical research in reproductive physiology there. But he joined our faculty in 2012, and Dr. Bridges currently teaches reproductive physiology. He gets to teach that every semester, and he's got about 120 or so students, I think, maybe a few more than that, I don't remember. But he's a very active a researcher, and Dr. Bridges is gonna tell you, give you an in-depth examination of his research in dietary selenium and its impacts on fertility and gonadal function in beef cattle. So with that, Dr. Bridges, I'm glad you could join us tonight, and I'm looking forward to your presentation.
Okay, thanks, Dr. Anderson. Before I start, I need to acknowledge Sarah Carr, one of my graduate students that you don't know, and you probably met Ben Cright. They're both doing their PhDs with me, and they have done the majority of this work. The second half of my talk has all been funded by the USDA, so I need to acknowledge them as well. Okay, so as Les said, well, we know we're a cow-calf state, about 1 million cows, 1 million calves each year. He talked about fescue. Now, we're not going to talk about fescue today, but I put this slide up because there is a lot of selenium fescue interactions that we're starting to explore here. So we're hoping to be able to also ameliorate some of the problems with fescue toxicosis using different forms of selenium. So our combined research program here at UK is trying to tackle both of these really major issues in the Kentucky beef industry area. Okay. As far as selenium goes, we don't always think about it, but we need to. It's basically an essential mineral. Now, an animal gets it from the forage. Foragers get it from the soil. So if you are a dairy farmer feeding silage that came in from another state, you may not have to think about selenium. But as far as beef producers go, your grazing all goes back to the soil. And throughout Kentucky, it varies by county. You can find out what your county's selenium content is on average, but we're fairly low, marginal to deficient in most counties in Kentucky. So we recommend supplementing with selenium. What selenium does is gets incorporated into what we call selenoproteins, and there's over 20 different selenoproteins that are identified. But selenoproteins are one of the body's major antioxidants, and we all hear about antioxidants. Whenever a cell is basically metabolizing, is metabolically active, it creates free radicals, they're damaging to the cell. So antioxidants, again, selenoproteins are basically antioxidants. They combat those free radicals, convert hydrogen peroxide into water, maintains the cell integrity, the structure of the cell. So especially with metabolically active organs like the reproductive system, these antioxidants are extremely important to maintain proper function. And this is just a quick visualization of the reaction that occurs within the cell. Hydrogen peroxide produced from basically the metabolism of cell, the expense of glutathione, which is a selenoprotein, gets converted to water. You're basically cleaving off that nasty part of that H2O2 to produce something that's not going to damage the cell. So that's really the overriding mechanism of how selenoproteins are going to work. What I like to put in these talks is what I have found as one of the best examples of how important selenoproteins or antioxidants are. GPX5 on the right of your screen here, that is one of those 20-odd selenoproteins, okay? Different selenoproteins are expressed in different quantities in different organs. So GPX4 on the left there is the major selenoprotein in the testes. GPX5, the one below there, is expressed in the epididymis. So as far as spermatogenesis go, which is where we're going to start this talk today, the epididymis acts to store and mature and concentrate sperm before it's ejaculated, okay? So what this group of researchers did, they took sperm out of, pardon me, a regular mouse, which is the one here. Wild-type mouse, looked at DNA damage in the spermatozoa, about 25%, normal amount of DNA damage. You treated that sperm sample with hydrogen peroxide, which we just said is what cells are producing when they're metabolically active, increased it to about 65% DNA damage. That sperm is not going to get an animal bred. Okay, and over on the right bar here, those mice were bred to have a deletion of one selenoprotein. That was the only difference in their mice. Out of that 20 group of selenoproteins, these ones did not have GPX5. The quality of that sperm sample from these mice was exactly the same as if you treated it with hydrogen peroxide. remarkable amount of damage that that hydrogen peroxide can do, which was the whole purpose of the. So we were just talking about the importance of the selenoproteins, and I brought up a figure from a mouse study that just basically showed deleting just one of these selenoproteins was the equivalent of treating your sperm cells with hydrogen peroxide. Okay, so commercially available, we have inorganic forms of selenium to supplement with, which is what most producers use. It's cheaper, it's readily available. Companies like Alltech are making an organic form. Remember that plants are producing organic selenium. Now the form, we've already been able to work out that the form affects a lot of stuff. It affects the bioavailability, how much selenium is in blood, as well as the bioactivity, what those selenoproteins are doing. What we often don't think about, it really changes the whole genetic structure of a tissue. We've looked at various organs. The pituitary stands out because it really controls reproduction. And it affects the whole transcriptome, which is a fancy word for saying all the genes in the pituitary that are making the proteins that get the job done. It affects the liver, and for my part of this, the liver is important. It's because it's the primary site of steroid catabolism. Now, the circulating half-life of progesterone, which is what we're going to talk about a lot today, is only about 30 minutes. Broken down very, very rapidly in the liver. So if we're affecting how the liver metabolizes progesterone, we're affecting systemic quantities of progesterone as well. Okay. For fertility point of view, we know the gonad is highly sensitive to these reactive oxygen species and therefore requires selenoproteins to maintain cell structure and function. Collectively, our objective is to figure out which form we give our cows to achieve optimal fertility in this case. Other people may be looking at different parameters. As a repro person, I'm trying to increase fertility. Long-term goal is to be able to say, look, you should be supplementing your cows with this or that or the other to get the best result. Okay, so this is a very applied, practical orientated research project. Okay. All the work I'm going to talk about today has been using selenium adequate animals. We know that if your animals are selenium deficient, you are going to have problems. Just about in every tissue, organ in that body is going to be affected. So none of the work we do at UK here is a deficient versus adequate state. It's all the different forms we can provide to maintain an adequate level of selenium for function. Angus cross cows, and we typically maintain our cows on either an inorganic form of selenium, which is the ISE there, an organic form, OSE, which is the Alltech product, or a mix, which is just a 50-50 split of inorganic and organic, okay? Equimolar amounts, all selenium adequate. Now, the first study I'm going to show you was looking at testes that were collected at birth from cows that were maintained on different forms. So all we did was castrate these bull calves 2 days after they were born, and we did what's called a microarray analysis, which looks at the whole transcriptional genome. We're looking at 25,000 genes. I was mainly interested in the genes expressed in the testes that affect either spermatogenesis or steroidogenesis. So we did this analysis, then some validation. Basically, our aim was to see what form of selenium through gestation is going to set up bull calves for optimal fertility. Okay. To orientate you in the testes, on the left is basically the functional unit. That round circle, let's see here, that's a seminiferous tubule. So sperm start developing as stem cells on the outside, and as they move towards the lumen of this seminiferous tubule, they go from a round cell, like up here, to a mature spermatozoa released into these lumens, okay? These cells on the outside here are Leydig cells. They're the ones that produce testosterone, which is required for spermatogenesis. Okay, so these two types of cells that basically comprise the whole testes, and they're working together to produce testosterone and viable spermatozoa. This figure in the middle, basically just the lumen would be down here. You can see round cells to the mature, Leydig cells out there close to blood vessels. And the reason I put them up is I want you to pay attention to this group of cells here. These are round spermatids. We're going to talk about those in a little while. To the right, you have a hand-drawn picture of a Sertoli cell. Now, these are the cells that basically surround and nurture the developing spermatogonia as they develop. And the reason I put this up here is because it was drawn in 1865, and I just think it's kind of neat that they did such a good job back there. Okay, so as far as selenium and testes function, we know the testes are going to develop in utero. There is some functional organization after birth, but the important thing is they become steroidogenically active very early after birth. Top, we have testosterone. Puberty is on that blue line, and you can see within, 2 months of birth, they're starting to produce testosterone. They're producing estradiol, though, very early after birth. We don't often think about estradiol as far as male fertility, but both testosterone and estradiol are required for male fertility. Testosterone is required for spermatogenesis. Estradiol is the main regulator of epididymal function. So the epididymis, again, that's what stores, matures, concentrates the sperm after it's been released from the testes before it's ejaculated. Okay. So we took these testes out within 2 days of birth, again, did a microarray analysis, found over 1,000 gene transcripts that were affected just by the different form of selenium that these cows were fed during pregnancy. All selenium adequate, all normal, all running around, but over 1,000 genes differed in these neonatal testes. Of those, there were 17 that were really interesting, and we're just going to touch on 2 for time constraints here. But one of the most important things about this slide is over to the right. This big, funny-looking figure here. It's called a PCA analysis. Blue is organic. Red is inorganic. Now, you would expect a 50-50 blend of organic and inorganic, that third treatment that we feed our cows, to sit somewhere in the middle. It stands out on itself. And we're seeing that in multiple tissues. For some reason, by mixing these 2 forms, we're stimulating uptake through different cellular pathways, and we really result in a different phenotype of the animal itself. Okay. This is one gene that I wanted to point out that's really going to affect Sertoli cell function. We said that Sertoli cells, these ones here, that basically house and nourish the developing spermatozoa. And I told you to remember these round spermatids. Well, they need energy, okay? The source of energy they use is lactate. A lot of cells use different forms of energy. Round spermatids, which are what those spermatogonia are called, require lactate. LDHA is the protein that converts pyruvate to lactate, okay? We have inorganic, organic, and that 50-50 blend. This top line here, this is a log scale, it's a microarray scale. Numbers don't mean anything, but this bottom line here, that's real relative values. This mixed form of selenium, there was 50% higher of the main enzyme regulating lactate, the energy source for these cells. So by manipulating that form of selenium, we're in turn provide to the testes specifically during the development of these spermatozoa. Okay. The second one, before we switch over to the female, is on the steroid part. Again, Leydig cells, those cells outside the seminiferous tubule, they're the ones producing steroid hormones, testosterone required for spermatogenesis. Gene we're focusing on the moment now is a different one, cyclin-dependent kinase 5. Again, if you look at that bottom, in that mixed treatment group, which is just a blended of the inorganic on the left and the organic there next to it, 50% higher expression of one of the main genes that regulate steroid production, okay? So we need energy, we need steroids. The forms of selenium together gave us a better response on both cases. When we look at the big picture, we start to look at the conversion between testosterone and estradiol. So remember, as far as steroids go, we're going to start with cholesterol. Cholesterol is going to be converted to progesterone. Progesterone is going to be converted to an androgen, androgen to an estrogen. They all tie together. But when we look at the different genes catalyzing all these steps, by manipulating the form of selenium, we can push testosterone towards estradiol. We can move estradiol towards the sulfated form. We can really change the ratio of the steroids that are then regulating gonadal function. And this is just in words here. Don't pay too much attention to it. The main thing is both testosterone and estrogen are essential for fertility. Although these changes we saw in the neonatal testes, that testes is structurally sound. You know, it's starting to make steroids within a couple of weeks. It's going to start making spermatozoa a couple of months later. But we're seeing changes that not only affect the testosterone amount or the total circulating level, but the ratio of androgens to estrogens that both drives testicular and epididymal function. All these basically lead on to acute changes that hopefully in the future we can tease out to give proper pre-breeding supplementation strategies for your bulls. Okay. Now, after we did this, I left the male side and got back to my roots, which is female reproductive physiology. Can we manipulate, just like in the male, can we manipulate the female side of the reproductive tract to produce, you know, optimum follicles, optimal luteal function, optimal embryo development, fertility, basically. Okay. So again, selenium deficiency, we're not looking at that here, but they've shown that it causes follicular atresia, ovarian degeneration. They've used inorganic selenium to stimulate the growth of follicles, estradiol production, progesterone production. There was a fair amount of suggestive information that this was really worth looking at. Most of the earlier studies are looking at a single form of selenium or using a single form because we know that we need that selenium. So here we're comparing forms in a selenium adequate model. Okay. This was the figure Dr. Anderson referred to from Dr. Fortune there. Just to give you a heads up on the estrous cycle, the top there are basically patterns of follicular growth. So this is an estrous cycle in a single heifer, 21-day average. You can see that there are waves of follicular growth. Okay, the little asterisks on the top point to ovulating follicles. Now on the bottom, the hashed line is progesterone, which is what we're going to talk about for the rest of tonight. So you're going to ovulate on basically day 1. That day 0 is the LH surge, which causes ovulation. A few days later, that corpus luteum starts to form, increases secretion of progesterone up to about day 15, 16. Now, what we're going to talk about today is that day 6, 7 on that upward plane of progesterone, okay? And then we're going to talk about day 17, 18, which was just before the decline of progesterone. And what we know that embryo, when it enters the uterus, it's going to enter the uterus around day 5, 6, okay? The level of progesterone at that time is going to affect the uterine endometrium and stimulate the release of the nutrients that are required for embryo development. So the higher the progesterone around day 6, 7, the better we're off. And they've known this for 30-odd years, but they've never been able to do it in a non-hormonal manner. It's always been artificially increasing the level of progesterone. And what we're going to see in the next few slides is the form of selenium can affect that day 6, 7 level of progesterone, which is then going to go on to affect that uterine endometrium to cause it to secrete the nutrients required for that embryo. That embryo is going to get strong enough to survive past maternal recognition of pregnancy, which has to occur before we have luteal regression, that progesterone drop, go on to implant, form placental units, and maintain that pregnancy. Okay, so here's a prettier picture of Johanne's last one with some nice pretty pictures at the top. So on the left up the top, you have a follicle, which is going to be producing estradiol, which is going to cause estrus. Now the cells of that follicle after ovulation are going to differentiate into this corpus luteum on the right, which is releasing progesterone. Okay. So we know that we have waves of follicular growth, increasing progesterone until we have luteal regression. Now with all the evidence that there was, I wanted to look at the follicle first. That was probably part of my background as well. And the best way to look at a follicle is to take that very, very first wave and transition it into a pre-ovulatory follicle. We get very good timing back. So if we move the time of luteal regression, we can cause that first follicle to go on and form a pre-ovulatory follicle. So we did an experiment where we collected pre-ovulatory follicles and looked at a bunch of different parameters with them. Very, very disappointing. Didn't show anything except one strange little happening, and that was this. Okay. On day 6, if you look at progesterone day 6, inorganic group 3.44 nanograms per mil, the organic group 4.5, the mix 5.14. That's about a 1.5 nanogram per mil increase in progesterone on a single day. Now, that's very, very easy to overlook. This was just a single time point. But that time point, which wasn't designed to actually show this particular time, is instrumental, okay? That's right at that time, as I said, when that embryo is needing progesterone-induced histotroph from that uterine endometrium. So this was the bit out of that whole study. That one line was the one thing that interested me, but no one believes a single data point. So I went on and repeated that the next year. Whoop, maybe I didn't. Repeated that the next year, did a few days earlier, a few days later. If you look on day 7 under that fall of 2015, again, 1 nanogram per mil increase, slightly different assay was used, so there's a slightly different level of numbers, but day 6, day 7, just by manipulating that form of selenium, we could increase progesterone at this critical time. Now, if we go back to that slide that I had before, again, that progesterone advances development of the endometrium. It's known to stimulate embryonic length and viability, overall increase in fertility. And there is honestly 30 plus years of people trying to do this in a non-steroidal manner. So we got very, very lucky. Now at the bottom, you'll see for that particular year, our conception rate to first service versus our overall pregnancy rate. Now we must have had a really, really harsh AI day that day. Could have been blowing we'll just say it was blowing a tornado because our conception rate to first service in the inorganic and the organic group were horrible, 30%. Mixed group was normal. So again, we have antioxidants that are affecting stressed tissues. And the only form of selenium that got our animals in this particular year up to an acceptable rate was that mixed blend of the two. If you look at the bottom, our overall pregnancy rate, again, this is after bulls were in for the whole season. We had 90% for all of our animals. So all of our animals get bred, but we get them bred quicker, especially this year where there was some stress going on when we had this 50-50 blend of organic and inorganic selenium. So that was the next one. And as proof of concept more than anything else, I then wanted to look at progesterone during pregnancy. Now, during pregnancy, there's typically 2 sources of progesterone. Now, progesterone is the one steroid hormone that is absolutely essential to maintain pregnancy. Now, most animals, the placenta makes a lot of progesterone. Cow, it doesn't, okay? In a ewe, for an example, you can take the ovaries out after about 50, 60 days and that pregnancy will be maintained. Women are the same. Horses are the same. Cows and pigs require luteal progesterone throughout pregnancy. So we saw again this mixed form maintaining a high level of progesterone throughout pregnancy. So it is really stimulating that corpus luteum. Okay. So what my role now is trying to figure out how it all works. So then I go to the lab, get my people to do the work, and I take all the credit for it. So we know that day 6, day 7 are those key days. We maintained our cows on these different forms of selenium. We got rid of the organic form actually because we wanted to utilize our cows more efficiently. So we're comparing the inorganic form of selenium to that 50-50 blend of organic versus the mix. Only thing different between the animals, the form of selenium they're being given. And on day 7, I took out the corpus luteum from their ovaries, okay? This was done down at Princeton, a pretty quick surgical procedure, popped that corpus luteum out. My grad students then started working and we wanted to look at progesterone secretion, some of the genes, blah, 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 a bunch of other things, and then get to hepatic function and blood clearance. Okay. Now, the good thing theoretically is the progesterone secretion by the corpus luteum is fairly straightforward. Start with cholesterol. Star protein down on that bottom left transports cholesterol to the inner mitochondrial membrane. P450 side chain cleavage is an enzyme that converts cholesterol to pregnenolone. Down at the bottom of the picture there. And then 3-beta HSD, 3-beta hydroxysteroid dehydrogenase converts pregnenolone to progesterone. So pretty straightforward path. And if you're looking at steroid production, you're going to look at all these 4 or 5 players, the associated receptors, and we should be able to pinpoint where the increase is coming from, because unless we know how this is increasing progesterone, we're not going to be able to use it to the best of our advantage, use that knowledge to progress things. So understanding that mechanism from my point of view is very interesting. So again, I looked at all those genes, and there at the top a bunch of them, and at the bottom a bunch of associated receptors, and it didn't really look at all promising. All those numbers show nothing except for that one in red, which was the nuclear progesterone receptor. Okay, so at this point of time, my graduate student's having a coronary because she thinks that the world's turning upside down on her. So we thought about it for a while, and it was like, if the steroidogenic machinery in that corpus luteum is not differing, why do we get an increase in progesterone that we've seen multiple years as well as through pregnancy? It's either going to be upstream or it's downstream. And it turns out it's upstream. Well, basically, cholesterol supply. Okay, so we know that cholesterol is the source of all steroids. So that luteal cell I showed you was just here, cholesterol to pregnenolone. So then we started backtracking through all the cholesterol pathway. Up the top here, there's a low density lipoprotein receptor, high density lipoprotein receptor. So basically, as far as cholesterol goes, there's 4 sources for a cell to make the steroids. We can uptake cholesterol from the blood via these low and high density lipoprotein receptors. Cells themselves can basically utilize cholesterol ester stores, as well as cells can make cholesterol de novo. And I haven't highlighted all the genes, but this pathway lit up, okay? So it wasn't the ability of the corpus luteum to convert cholesterol to progesterone where we were seeing the effect. It was the ability of that corpus luteum to either get cholesterol out of the blood or the cell to make cholesterol itself. So we finally got a handle on why we can see that increase in progesterone. And it wasn't from the natural starting point, it was a little bit upstream. The other interesting thing that gave my student, who just texted me and said that I was throwing her under the bus here, was we cultured these cells. Now, Sarah took these luteals, corpus luteum, she dissociated the cells, very kind of complicated method. So I was happy to let her do it while I played with the cows. Ran them out for 4 or 5 days in culture and looked at progesterone secretion. Now, I've just told you that the mixed group have higher progesterone. Now, the mixed group on this figure here, which I failed to label well as these light grey bars, lower progesterone. The black bars were our inorganic group. Now, that goes against everything that we thought was going on until you think back to that cholesterol. Do they have the substrate? But probably as important to that is within that corpus luteum itself, there's 2 different cell types. there's large luteal cells that are derived from one part of the follicle called granulosa cells, and there's small luteal cells that are derived from the theca, which is the other steroidogenic part of the follicle. Small luteal cells respond to HCG, which is human chorionic gonadotropin, the same thing basically as luteinizing hormone. Large luteal cells respond to things like prostaglandin E, growth hormone, stuff like that. So in this particular case, it looks like the small luteal cells in the inorganic group had enough cholesterol to drive that progesterone further, which makes a little bit of sense because generally they're not producing the majority of the progesterone by that corpus luteum. So although some of these results look a little funky, we can figure out what it all means in due course. Okay. Now we then did what's called a bioinformatic analysis. I ran or my students ran another microarray, and again looked at all the genes together. Now all the green little dots at the bottom are the ones involved in cholesterol biosynthesis that we talked about. Always keep in the back of your mind that this form of selenium is affecting the antioxidant status. Antioxidants, major immune regulators. And we actually have a direct tie in the middle between cholesterol and immune function that is all being affected just by the form of this mineral they're eating. Okay. What we're doing next, which we started this year, but the old COVID put a kibosh on the whole thing. So we're doing the same type of experiment, but rather than taking out those corpus luteum on day 7, we're going to breed the animals and slaughter them at maternal recognition, and then we can look at the conceptus itself, the endometrium, the luminal fluid, the liver. Now, day 17 is the time of maternal recognition of pregnancy. So that is when the conceptus is signaling to mum that it's there and it needs that corpus luteum to be maintained. So that's then going to tie this together. We'll have a full understanding of corpus luteum. Then we take that forward 2 weeks into a pregnancy. We can see what that change in progesterone is doing to not only the endometrium, but the embryo itself. And after that, we should have a really good handle on how we can manipulate these forms to optimize fertility there. So just as a quick take-home message, we know we need to supplement with selenium. All the work we do, selenium adequate. We've seen a bunch of parameters affected by the form of selenium, including both estrous or early luteal phase progesterone, pregnancy. We've seen prolactin changing. Didn't talk about prolactin today, but you'll get a earful of that in one of the upcoming talks. Every tissue in the body we've looked at is affected. It's been a remarkable 5-6 years of research, actually. And the hope is to be able to say, look, this is the best way to get those first service conception rates up, overall fertility up, so that producers get the best bang for their buck and can make the most money and keep the kids happy. So with that, I think I just have to again acknowledge funding for the female side of this was supported by the USDA. We've got a couple more years in that grant, and hopefully we'll have a good story to tell by the time that's all complete. Thank you.
Great job, Dr. Bridges. If anybody has any questions, please type them in the chat. And Dr. Bridges will handle the questions as they come in. Phil, I'm going to start off just to make sure that everybody's clear. The form of selenium in the research that you are doing is not changing the genes, it's changing what the genes produce. I just want to make sure that everybody's aware that we're not changing the genome, we're changing the transcription of certain genes and the products of those genes, and those products are what are impacting the physiology.
Right. More just the level of the product. So for say that energy one, the gene was being stimulated to produce 50% more of the protein that was required for the energy. So yeah, you're not changing the gene at all, you're just changing how much that gene is telling the cell to turn into the protein to make.
I just wanted to make sure everybody was clear on that. I mean, what I think is a vital separation. Now, one of the reasons that I think the population stuff that you and Dr. Matthews are doing is vital is ultimately you could see some epigenetic changes in gene expression associated with long-term exposure of populations of cattle to, form of selenium, and I think that'll be fascinating as well.
And that's very true. As when we get a good handle on this first study, and again, these studies take 5-10 years, but epigenetics has been on our radar now for a couple of years, and we would really like to be able to, if this work here is successful, then I may be able to get the funding to start looking at epigenetics, because I am confident that we're going to see some epigenetic changes as well.
Yeah, I am too. Daniel Cummings asked the question, have you evaluated any effects of strategic administration of inorganic injectable trace minerals on gonadal function that you discussed?
Short answer, no. Long answer is we have performed a preliminary study on adult bulls where we looked at sperm quality, and that was comparing the mixed to the inorganic. We saw some effects, but we didn't have the bull numbers to actually draw any real conclusions from it. So even with nothing acute, no injectables, but even by manipulating this, the mineral fed, we have tinkered on the bull side, but we didn't have the number of animals to really draw really quality conclusions from it.
One thing I wanted to add to your fertility discussion as far as the cows down in Princeton goes is, of course, you picked the 1 year, right, where we get 30% conception rate to AI. If
you're going to ask me to do this, I'm going to throw out that year's data for you.
yeah, I hear you. But to follow up, I mean, we've synchronized and AI'd those cows for many years, and every year the organic form is higher than the cows on the consuming the organic form. Their conception rate to AI is higher than cows consuming the inorganic form every year. And this year, and I told you about this the other day because Ben and I went down to breed those cows, and every single cow that was in the group that was consuming inorganic, so the sodium selenite, they all had been in heat when we were breeding them, because we had the patches on and we had the electronic heat detectors. But only about 60% of the cows in the organic form, in the sodium cellplex in the group, were showing estrus. But we had a 9% increase in conception rate to the cows consuming the mix. And so it's and you know, and those are normal conception rates to AI every year except the 1 year you threw up there, right? Those are normal ranges for conception rates to AI, and it always exceeds the inorganic by at least 5%, at least.
Yeah, yeah. Actually, I had the following year up as well. Well, I didn't put it in this because it wasn't quite as outstanding, but the year after that, I think it was a 15% change in first service conception rates. It was like 45 to 60 that year. So
I don't think we've ever gotten less than 60% in the mixed group, and we rarely get 60% in the selenite group.
I'll agree with that. Yep. Yep.
And that's many years of work. I mean, it's not 1 year in time. It's every single year. The organic form exceeds the inorganic. Kevin Laurent asked us to define epigenetics.
So it's basically a carryover effect. So really passed from parent to the next generation, okay? So rather than what we talked about today was just transcriptional, it's just how much that gene is changing. Epigenetics, you're passing stuff to the next generation, you can change the actual gene structure itself. So there can be more long-term carryover effects.
There, you want to chime in on this, any?
Who?
To figure out how to get off of there. Yeah, I mean that's basically right. It's an environmental effect now, basically, that is going to have a effect on gene expression later on. So how I would explain it.
One of the things that's always fascinated me about epigenetics is we don't realize that the environment the animal is raised under can actually impact the gene expression of oocytes, which are eggs, and sperm cells. And so it ends up, you know, we don't really think about that at all. And so that's really what epigenetics is that long-term impact of environment. So, and how that it changes gene expression. Dr. Bullock just threw up the screen next week. We're going to have Dr. Dave Harmon and Kevin Laurent are going to talk about nutrition. Dr. Harmon has been in the Department of Animal Science for many years and is a internationally recognized new ruminant nutritionist. And I'm sure he's excited to share his research with our clientele, our producers. And you'll be able to find this and all of our Reaching Out While Locked In webinars on our YouTube website. And just go, if you didn't catch everything, or if you wanted to go back and review some of what Dr. Bridges talked about tonight, just pull it up and review at your pleasure. Phil, any last comments?
No, I thought that worked very well. If anyone wants to get in touch with me personally, you can find me on the UK Animal Science website and shoot me an email. If you need to follow up with anything like that, I'm more than happy to answer questions as well.
Dare, Jeff, Katie, Kevin, any comments before we conclude?
I'm good. Appreciate it, Phil.
You're welcome.
Thanks, Dr. Bridges. It's a great overview.
You're welcome.
With that, we'll conclude this seminar on form of selenium and fertility, and thank everybody for tuning in. See you next Tuesday.